Types of media include liquid broths, which are good for growing large quantities of culture. The drawback with liquid cultures is that they don't show morphology. And that's going to be like the key characteristics that help you identify what an organism is. Solid media, on the other hand, different augers like auger plates, auger deeps, and auger slants, those are really good for highlighting key characteristics. Those key characteristics include things like, is it able to grow or not? Because some media are inhibitory to certain groups. You can also see color. Some media have special ingredients that promote a color change for certain groups. Other things that can be observed are the colony size, shape, and texture, along with the odor. Microbes vary in their nutritional requirements. No single medium supports the growth of all microbes. Defined medium have known chemical compositions, and all of the amounts of ingredients are known. A complex or undefined medium is chemically undefined. It contains parts of plants or animal digests or yeast extracts. These are widely used in labs. Nutrient media provide basic nutrients to support growth. Most groups grow on nutrient media. These media, however, don't usually highlight key characteristics. They're basically just a surface for the organisms to grow on. Examples of nutrient media are nutrient agar and blood agar. Most organisms will grow on these media. Enriched media have special nutrients to support the growth of fastidious or picky organisms. Only certain groups will grow, and sometimes even only a single genus. Enriched media may highlight key characteristics. An example of an enriched media is chocolate agar. Chocolate agar has red blood cells just like blood agar, but instead of being whole red blood cells, these blood cells have been ruptured or broken open so that the fastidious or picky organisms can get to the inside of the blood cells. So organisms that can grow on blood agar are able to open those cells themselves, but these fastidious organisms, they can't get inside the blood cells without help. So they won't grow on blood agar with whole red blood cells. Instead, they grow on chocolate agar because the chocolate agar has ruptured red blood cells and those nutrients are open and exposed and available to them. Other examples are Bordeaux media that's special for Bordetella species, brilliant green agar that grows Salmonella, citramide agar for Pseudomonas, Fletcher's medium, and Loeffler's medium. There are many, many, many other types of medium, each with a often very specific type of organism that they will grow. Selective media have growth inhibitors. So media is selective because it is able to inhibit the growth of one type of organism while allowing other ones to grow. Examples are McConkey agar, which will inhibit gram-positives but will allow gram-negatives to grow. Another example is mannitol salt agar, which will inhibit gram-negatives but it will allow gram-positives to grow. Eosin methylene blue agar is similar to McConkey agar in that it encourages gram-negatives to grow, but it doesn't allow for gram-positives. These same media are also differential. A differential media contains dyes or some type of indicator system that allows it to highlight differences between groups. So the McConkey agar allows gram-negatives to grow, and then it has a special sugar in it that will when fermented, the colonies turn pink. So organisms that can ferment this sugar show pink colonies, while organisms that don't ferment the sugar show clear colonies. Similarly, mannitol salt agar has a special sugar. When fermented, the organisms grow yellow. If they are not able to ferment the mannitol, then they grow clear red, or red, the color of the media that they're on. Finally, the eosin methylene blue agar will allow organisms to grow and then turn this sort of sheen color if they're allowed to ferment a sugar very quickly. And if they don't ferment, then they have just a regular sort of clear sort of appearance.